we're going to be moving into our next set of activities here, which is this topic area 2. And the title of this one is going to be herbicide resistance in weeds. So you'll find a slide set here. The actual module is here as a Word document. The student answer sheet can be found here as well. And then I've posted the 2 articles that are referenced within the module in the event you want to download those separately. I'm not sure what your access is, particularly to that peer-reviewed paper, but that PDF is there for you to look at. And so we'll spend a little more time on that later, but we're going to go through the slide set first. I just wanted you to see where it was in case you wanted to follow along with the materials as we were going. So we're moving into our second section here on herbicide resistance in weeds. And I've got a few focus questions for us here, and I'd like to have a little conversation about these 4 questions. Particularly, why are weeds considered pests? What herbicide resistance? This should be what is herbicide resistance? Where does resistance come from, and why are people concerned about herbicide resistance? So let's go ahead and start with that first question. Why are weeds considered pests? This may seem like a fundamental question, but it's good to think about every now and again some of these basic questions.
Yeah, so we've got in the chat, Heather says that they take nutrients from the other plants, and Courtney says they take up the space of the plants we want there. Yeah, thank you.
Yep, so they compete for things like nutrients, space, light. You wouldn't really think about light, but they do. Oftentimes our weeds have a faster growth rate than our crops. So that can be a concern as far as their competitive ability. Another one that we don't usually think about in Ohio in particular is water. When we're thinking about out west, where we're in more arid type environments, having as much water for your crops to grow is really key for minimizing yield disruption. Here in Ohio, we get a lot of rain, so we don't usually think about water as being too limiting, but in dry patches or dry stretches, it can be a problem. So yeah, they essentially take resources away from the plants that we want to be using those resources. We want specific plants to use the light, use the water, use the nutrients, and not have it be taken by some other species that we're not trying to grow. So that's why weeds are considered pests. Yes. What is herbicide resistance? Maybe I should have started with another question of what is a herbicide? So what is a herbicide and what is herbicide resistance?
Courtney adds, the weeds just consider weed killer a spot treatment. That's
in the event of herbicide resistance. Okay.
Yeah.
Yeah.
Herbicide is a
chemical that kills plants.
Chemical that kills plants. Yep.
Yeah, that's essentially what it is. So a herbicide is a type of pesticide that's applied to kill specific versions of plants. Some herbicides are very specific. They'll only kill certain kinds of plants because other plants can metabolize them or have a different metabolic pathway to work around where that herbicide is actually acting within the plant's metabolism. Sometimes it's a physical disruption. Some of our organic herbicides like vinegar or acetic acid, they essentially just, it's like pouring acid on something, right? It melts it. It's not a good situation. So sometimes it's a physical disruption like that. Other times it's a chemical product that's used to manipulate some part of plant's physiology. And so when we think about resistance to the herbicide, it means one of 2 things. It either has an alternative pathway built into its biology, so it can continue metabolizing whatever's trying to be stopped by the herbicide, or it has the ability to break down the herbicide. So many people had mentioned GM studies in their classroom or working with transgenic plants or GMOs. Most of our GMOs to start in corn, the first GM crop to come to market was one that had a Bacillus thuringiensis gene inserted into it. Bacillus thuringiensis is a bacterial organism that is actually labeled for use in many organic production systems for control of insect pests. And so what they did is they took the gene of interest that makes Bacillus thuringiensis a good organic pesticide and inserted it into the corn genome. And so it codes for a new protein that corn wouldn't normally create. And so that was kind of the first GM trait. It was actually for pest resistance against lepidopteran pests. And there was another trait that was inserted for coleoptile or coleopteran pests, beetle larvae. So the one was for corn borer resistance in the stems. The other was for corn rootworm resistance. In soybeans, the first GM crop that we had come to market was actually Roundup resistant soybeans. Roundup Ready soybeans or glyphosate resistant. Glyphosate is a broad spectrum herbicide. And the first one that came to market was one that could tolerate application of that glyphosate. Instead of killing the soybeans like it used to, the soybeans would no longer be killed by it. The first GM trait inserted was a modified version of the enzyme that the plant used to make the amino acids that were disrupted by glyphosate itself. And so that was our first instance of crop resistance to a herbicide. And it was so effective that within a few years of coming to market, the vast majority of soybean acres were planted to this resistant version. Pretty soon after that, it came into the corn as well. And we started to see glufosinate resistance come in, which is another broad spectrum herbicide with a slightly different process. But that's kind of what herbicide resistance is. It's the ability to tolerate a chemical treatment that would otherwise have killed that plant. Okay, so in our crop plants, we did it intentionally through GM modification. Those initial GM traits never actually increased yield in those crops. They basically made them more resistant to a pest so that the feeding wouldn't hurt their yield. And it made them able to have better weed control in their fields so that when you spray the one application, you're weed free for the rest of the season. And so then yield increased because of that. The traits didn't actually increase yield. It just made management a little bit easier within those crops at that given time. And so resistance is the ability to not be affected by the herbicide. And so where does resistance come from? I mentioned where it comes from for some of our crops in the case of glufosinate resistance or glyphosate resistance. Those would be things like Liberty Link soybeans or the Roundup Ready is another one. We have multiple more versions now on the market. We have dicamba resistant soybeans. We have 2,4-D resistant soybeans. And some of those genes insert into cotton as well, which is another dicotton crop. We also have Clearfield corn as another example. It's resistant to a herbicide called, I think it's called lightning. Kind of an interesting situation. But those were inserted genes intentionally. But where does resistance come from in natural populations?
Mutations. Thank you, Becky.
Mutations?
Horizontal transfer, William ends.
Okay. So I think Becky kind of hit it there on the head, but the mutation doesn't necessarily pollution. The mutation that we see in natural plants doesn't come from the actual herbicide itself. The herbicides themselves are not acting as mutanogens on weeds or weed populations. When you spray it, it's not like a Marvel comic, right? Like it's not like, oh, I was treated with glyphosate and now I'm a super weed. It's not manipulating the genome in that regard. But through the processes that Wendy described that we leverage in our favor for breeding plants, looking at allelic variation, different versions of genes that exist, that's essentially what happens in natural populations that are treated with herbicides. We have a broad spectrum of alleles that are present in a naturalized population of a particular species. And when you start applying a herbicide that kills the majority of those biotypes, it essentially applies a selection pressure to specific alleles that allow plants to resist dying from that herbicide. And so it's selection pressure that selects for particular individuals in that system that can tolerate that material. And so essentially, it's kind of like natural selection. There's natural allelic variation. And what we're doing is we're artificially accelerating the process of selecting for particular individuals. And so the last question there is, why are people concerned about resistance? This kind of comes back to our connection with ag, right? When we think about resistance and why we care, is because herbicides are one tool in a suite of toolbox for agricultural production and productivity. When Wendy was talking about DNA being discovered in the 1950s, my head initially went to, that was when 2,4-D came to market. That was one of our first synthesized herbicides. It came to market right around that 1945, 1950 range. And so that is when we started to see herbicides come to market. And they've been a very popular tool amongst producers because they've been able to optimize production and minimize competition without having to do things like intensive tillage, cultivation, hand pulling weeds. So they're a very attractive tool because they can actually help reduce labor needs on farm. But as resistance starts to come about, it means that tool is no longer effective. And so we lose a tool in our toolbox that has been effective for a long time. So Shelby mentions continually producing new effective products as herbicides. Some could be more harmful. Yeah, that's an issue. We don't want to release a new herbicide that is more potent or more damaging than it has been in the past. And so there's actual restrictions now on what chemicals can come to market based on what we've learned. We know, we've read Silent Spring, most likely. Most of us are familiar with Rachel Carson's book about DDT and the implications that had on our ecological food systems with bioaccumulation and an unintended consequence of using that product. And since then, there have been multiple safety layers in place to say pesticides can't last that long. They can't be that broad spectrum. They can't be that damaging. They need to break down, and they need to be relatively safe if ingested by animals and people. And so because of that, the products that have been coming to market since then have become less harmful, less long living, but it also means that the ones that we have, we need to be able to protect the use of for as long as possible. Because it's more and more difficult to create things that are effective, but still meet all of those other restrictive criteria. And so preserving the tools that we have is extremely important, both from an environmental stewardship perspective, but also from a practical production standpoint. And so that's why we're concerned about herbicide resistance. Yeah, I appreciate us going through that. The slides here basically hit on what we've already talked about. Weeds are considered pests because they rob crops of resources like water, light, nutrients, and space, and they usually limit crop yield. Another issue that we think about with why weeds are considered pests is because of their relative density. If we think about how thick a field of soybeans is planted or a field of corn, we tend to plant for corn to maximize yield somewhere in the range of 30 to 36,000 plants per acre or per football field, right? That's what an acre size is. But in some cases in those fields, if we did weed counts, if we took a square foot and we counted the number of weeds in that square foot, we could have anywhere from 10 to 25 weeds in that given foot if we just let them come up. And so if you scale that up to a acre basis, multiply 10 by 43,560, because that's how many square feet are in an acre. That gives us over 400,000 weeds in that particular area, compared to those 30,000 corn plants. It's a lot different of a ratio. And so there's a lot of weeds versus crops in any given system. So having an ability to reduce the pressure from those natural populations is key for agricultural production. Another issue with weeds is they could be alternative host for diseases, insect feeding as well, meaning that just their presence could increase the population of a pest that could be detrimental to our production systems. So having control of those species is really important. Things that impact weeds as pests are things like the life cycle, when they grow, how fast they grow, what's their ability to come back. Back in session 1, we compared seed production for different species and how quickly some of those species would overpopulate the system based on how many seeds they produce. Some plants like giant ragweed produce like 5,000 seeds. Things like water hemp or our amaranth species can produce 500,000 seeds per plant in a given year.
Yeah, just per plant.
That's per plant, yes. So it can be very easy to get out of hand really quickly. Weeds can come in on equipment. That's oftentimes how new species are introduced to a field, is a seed gets transported from one field to another. And it could be a problem there. Sometimes we think about growth habit, how they grow. Are they prostrate growth, low growing? Are they growing upright? And also, what are the growing conditions? If the growing conditions tend to favor the crop, they're going to favor the weeds as well. So when we think about this, we need to think about herbicide resistance in weeds. And so when we think about herbicide tolerance, it's the inherent pre-existing ability of a species to survive and reproduce after herbicide treatment, not caused by selection pressure. Weeds have a natural variance in this ability. I mentioned the first Roundup resistant crop was soybeans, and what they used to facilitate that resistance was a modified enzyme shape. They found an enzyme that could still operate, but wouldn't be connected to glyphosate. So the plant could continue to make the aromatic amino acids without being affected by the pesticide. And that slight change in protein shape comes because of these slight mutations that we see. These point mutations code for a slightly different amino acid that still allows the enzyme to retain its function, but it just has a different shape. And so then the way glyphosate would interact with it would be slightly different. And so it's not caused by selection pressure, it's just caused by this crossing over, right? Not John Edwards seeing ghosts. It's like, oh, the chromosomes change a little bit, and there's these point mutations that occur just from natural occurrence. So herbicide resistance is the acquired heritable ability of a weed population to survive a herbicide to which it was previously susceptible, and that's caused by selection pressure. So what does this look like? We can watch the evolution of herbicide resistant weed population through kind of a demo like this. Essentially, we see a wide range of genetically diverse weeds, especially if it's an outcrossing species. Remember, when you have an outcrossing species, it's a lot easier to get new combinations of different alleles in any given generation. If a plant is self-pollinating, that evolution of diversity in traits is a lot slower. And so it occurs at low frequencies within a weed population naturally. The herbicides themselves do not cause the mutations, okay? And so what we see here is this version here, this red version, just has a slightly different biotype. And so how we get this evolution of herbicide resistance is that over time, we see a change in the successive generations because of the selection pressure being applied. So in year 1, the herbicide works really well. It kills everything except for that red R. But then the only one that's left to produce seed is that red R plant. And so it'll produce seed, distribute those, and then we'll find more and more of those plants throughout the field that exhibit that resistance trait. You apply that herbicide again, and it may not work as well. Then you see that continue on, and before you know it, we have this problem. It progresses essentially logarithmically. And so it can go from being not really a problem to being oh no, what happened here? Aliens distributed seeds in my field and now they're all resistant
Mhm.
within a very short time frame. So this is demonstrating that. Not only is it showing, I like this diagram because it shows the plants themselves. The red one again is resistant, but it also shows the resistant seeds in the seed bank, because not every seed that a plant produces is going to germinate that first year. And so these plants will end up living for a decent amount of time in the form of the seed bank. And so eventually more of those will start to germinate, more seeds will be present in the seed bank. Continue to go, control is still very good. You see some escapes. Eventually, application is going to start to not be as effective. And then you have herbicide resistance, chemical application failure, and inability to even harvest the field. It can't be reversed in a practical time frame. In many cases, the seed pool is unlikely to change back because there is no fitness penalty to having these plants present. This is the story of what has happened in many parts of the South with Palmer amaranth and soybean production. It gets to the point where they grow a crop of Palmer for 3 years in an effort to draw down their population. When we think about how long a farmer's lifespan is and how many years they have to actually farm a piece of ground, that's about 10% of their productive potential. You lose 3 days or 3 years of production, that's 10% of the longevity of that field for that production environment. It's kind of a big deal. And so we want to avoid it if we can.
You're looking at the chat, the good graphing exercise, and then example of directional selection.
Sure, yep. Yeah, those are both very good points, yep. And so the activity that I have here today, there's a few different levels. There's kind of a two-part component. There's one where we have a few videos. There's a YouTube channel put out by some people called Ag PhD. By and large, I'd say about 80% of the material is pretty good. The other 20% I question personally. So sometimes it's not like everything they say is the 100% truth, but in general, it's a good resource that you can look at. If you have questions about accuracy of videos, if you find one that you might want to use, feel free to send it to something like me or Wendy, and we can take a look and can provide our insight as, you know, this is pretty good, or it's best to stay away from this one.
Yeah.
But I did find one video that I felt pretty good about sharing with you today and having you look through. There's another two-part video series from our OXU Extension weed scientist, Dr. Mark Laux, talking about this idea of herbicide resistance in pigweed populations, particularly water hemp in Ohio, and how it becomes a problem and how to try to keep it from being a problem. And so that's kind of the first set, is watch those videos and then answer those reflection questions. The second set is a little bit more of a deeper dive. It might be for upper level students, maybe higher level in high school. The one is actually looking at a peer-reviewed journal article to look at how weed populations change over time in a cotton production field. So it's not necessarily relevant directly for Ohio from a crop perspective, but it does give some insight into the spread and how those seeds move through the field.
And then there was a comment about this being very applicable for South Alabama and Georgia.
Right.
So yeah, definitely throughout
It's a
the
lived
nation.
experience
Mhm.
potentially for some of your students or
Mhm.
what you've seen firsthand. So it's definitely applicable. The other one is a CSA News Magazine article that talks about how to leverage some of the newer genetic tools in combating herbicide resistant populations moving forward. That's a little different than having wagons, hand laborers pull all the Palmer, put it on the wagons and then burn it repeatedly.
Mhm. Very labor intensive.
Because that's what happens. That's how you get these fields back into production. Are there any questions or comments about what's been presented? I appreciate the feedback of the lived experience from some of our participants seeing this happen. Does this make a little bit more sense? Does this change your viewpoint on what herbicide resistance is and how it develops? Again, when we think about this term called frankenweeds,
Yes.
this is something that we see when a weed becomes resistant to more than one herbicide. And it's really easy for hybridizing species or outcrossing species to get to the point of becoming frankenweeds, because it's like, hey, I'm a parent and I'm resistant to these 2 types of herbicides. And then here's another parent that says, oh, I'm resistant to these 3 herbicides. Let's get together and make some seeds. And then they come together, and the alleles that resist both of these and all 3 of these come together. And then you have a 5-way resistant species. And so it gets out of hand really, it can get out of hand really quickly. And so it's very difficult, and it's a very real problem. But it's not because the herbicides themselves are causing the mutations to occur. The herbicides have met all safety standards. They're safe for people to use as long as they're using the PPE that's outlined in the label. But the selection pressure, the directional selection, is what we're talking about here as far as why resistance occurs.
And for upper level students, you can talk about some of those different modes of action, because what we're talking about is, we've kind of mentioned 2 or 3, but there are multiple herbicide classifications that have various modes of action trying to, quote, attack the metabolism of the plant, or as Alex said, to, try and kill them by, just being surface applied. But I don't think that there's a lot of talk about how those actions of different herbicides work. And so that if you're really got some students who are interested in those pathways, there's a lot of really good information about how those pesticides actually do their job.
Right. Yeah, it's really amazing. The lightning herbicide I mentioned is one that it affects the ACC-ACE enzyme, which is one that affects fatty acid metabolism. And so we know that fatty acids are needed in every plant with phospholipid cells. And so if you impede the production of fatty acids, you can't make more cells.
Right.
It's game over.
Well, and there's several that attack different photosystems throughout the photosynthesis pathway.
Yep.
So it might give your kids a reason to understand what photosystem 1 and 2 are when you have to cover that in biology, that there are actual people who have, figured out this system so well that they are interrupting it so that plants will die.
Right. Like ferredoxin giving its electrons to NADP plus to form NADPH. There's a herbicide called paraquat that comes in and steals those electrons and then just releases them, causing cells to lyse. It creates free radicals and creates craziness within those cells. And like Jane said, there's 3 different types of photosystem 2 inhibitors that specifically affect that one protein. So it's really incredible the specificity of some of these herbicides, but it's also easy to understand then how just changing the protein slightly affects the ability of that herbicide to work. And so in this activity, students will learn about the resistance and how it works, and learn how human activities can influence the spread of populations. So this is the module 1. This is the teacher handout that I'm showing here. It has these materials. Jane had mentioned this idea of the herbicide modes of action, and this is a poster that shows the different 11 biggest resistance threats to crop production. Some of them you might not think are that big of a deal, but they can be a big deal. And the numbers that are corresponding here show the types of herbicide resistance that have been documented in some of these individuals. This isn't meant to say that every Palmer amaranth plant that you see is resistant to every one of these herbicides, but we know that resistance to these herbicides does exist in a subset of these populations. Not saying all of them are resistant to all of them, but sometimes these herbicides don't work anymore on those particular species. And so that's where we see that information. This I will take action chart can also help to tell you what these different sites of action are and what products are related to those if you want to take it a step further. I think one of the extending activities from Grow Next Gen also incorporates this idea of mode of action and efficacy in the field. And so we'll talk, Jane will speak more on that a little bit later. But just to give you an idea of this reference chart here, what we're looking at. And then there's these two videos here. The second one is about this idea of human mediated influence. So the PDF or the link here goes directly to the actual website, but I have the PDF available for you for use. But it's a weed science article that talks about infield movement of glyphosate-resistant Palmer amaranth and its impact on cotton lint yield. And I've tried to be very specific with this activity, saying, look in this section and see what details you can find, okay? And so that's another one of the activities. The last one is this genetic approaches to weed control. So it takes this idea of how do we get herbicide-resistant populations back on track, but use modern tools if possible. So that's what this article is. So what I'm going to ask us to do is for the next maybe 15 minutes is choose to do one of these activities. Either do the weed resistance videos and answer those reflection questions, or look at the cotton article from Weed Science, or look at the genetic approaches article. So either do the videos, the one peer-reviewed paper from Weed Science, or the genetics magazine article, and go ahead and spend some time and actually work through that. Read that article and see if you can answer those questions. Both Wendy and I have provided kind of what anticipated answers are going to be in italics. So try not to work off of that sheet. Please look at the student sheet instead. So again, you can find this on our AgStem file here. The news article is here. The peer-reviewed paper is here. And the video links are embedded within the student answer sheet. So go ahead and take some time and work on this activity, and we'll reconvene at about 11, sometime between 11:15 and 11:20. Okay, so coming back together, how many of our participants elected to do the videos? Either by show of hands, thumbs up.
Shelby has her hand up. And Joe has a thumbs up. Heather. Yeah.
Okay. So how many elected to do the paper on the spreading?
Becky,
Kelly? Jonas?
yep, William,
Okay. And how many did the genetics article? I see Becky with that one. Anybody else do the genetic tools one? Okay, so how about the video? What did you think of the videos? Did you think those were useful? Could you see yourselves using those in the classroom? Was it clear enough what was being discussed, or were they too specific? Feel free to type in the chat, unmute.
For the videos, I definitely thought they weren't too complicated for like the 11th, 12th graders. I would probably just modify the question, maybe to separate them.
Okay, Shelby, if you were trying to speak, I don't think we could hear you, but
Okay.
no, still not hearing you.
are we muted? We're not muted,
I don't
right?
think we're muted. So maybe, is our audio working? Shelby, you want to try it one more time?
Okay, so you can hear,
They can hear her.
Okay.
oh, her and us. Oh,
Okay.
yeah, that's fine. You can type.
Okay, yeah, we're having issues with our system here with feedback, and so Shelby, if you want to try again, I think we can hear you. Okay, shorten the questions for IEP. That makes sense.
I put it in the chat too. I don't know if you can hear me now.
Yep, I got you now. Yep.
Okay. Yeah, I just said that it would, like, the videos were good for like 11th and 12th. I might just like modify some of the questions just to like break them up a little bit, but I thought it was good for 11th and 12th.
Sure, understandable. Yeah, it's kind of an interesting situation with the herbicide resistance and what you can do if they end up starting to become out of control. It was interesting to hear this day and age recommendation to pull the weeds by hand. So yeah, the scientific paper, I think that's something that sometimes I struggle with as a college professor, of having students read through articles and really synthesize it. But it's a really good place to look at cutting edge research, kind of first report of these phenomena, and seeing where it comes in. I jumped right into the materials and methods because I was super excited about this paper. And then GR, I just couldn't understand what GR was. I was like, is that a type of, what does that even mean? But then I realized I should probably start at the beginning. And so I read the abstract, and that's where GR came out. Does anyone want to share what GR means in this particular paper? What does the GR mean?
Glycophosphate resistant?
Yes, glyphosate resistant is what it means in the context of this paper. And so what these authors did is they took these glyphosate resistant seeds and they purposely put them in a new field, because they're like, Palmer's everywhere. These fields don't have Palmer yet. So we might as well do it on our terms and get some research data out of it. And so they were able to put those in one end of the field, and then they just let them grow. And then they tracked their emergence throughout the field in future years. And so I thought that was kind of an interesting story. And that's what the figures end up showing in that paper, is where those seeds were going, where they were starting to appear. And why did the researchers say that they were moving? Was anybody able to pick that up?
Rain.
It was moving with rain in some instances because they broadcast these seeds on the surface. They got heavy rain and they started moving. But cotton production is interesting because they grow it in beds that are cultivated to be raised. And so they can furrow irrigate, put water in between the beds and then irrigate that crop. And so the weeds didn't move laterally. They didn't move sideways. They moved in the direction of the beds. Also, if it wasn't because of rain, it was because the combine that was present at the end, or the cotton gin that was moving through and actually picking the cotton. It's not a gin, it's a cotton picker. The cotton picker was moving in the direction of the beds. And so the primary spread of the weeds was along the beds as opposed to laterally through the field. And I think that speaks to how human mediated movement of seeds can be tracked and shown, and how it differs from how plants might do it in a natural ecosystem. And so this field started to get infested because the grower wanted to harvest cotton. And what happened by 2010? Did anybody find that in the article? Essentially, by 2010, the infestation was so great in the field, they were not able to even harvest cotton. So there was no yield data from the last year. And so they introduced the crop of pigweed in 2008. And by 2010, they couldn't even harvest the field. That speaks to how quick this problem gets out of hand in particular cases. So that was the article. Did anybody do the genetic approaches? So with this one, it was focusing a lot on the pigweed species. That's kind of where we've been, because pigweed is a notorious outcrosser. And so that's one of the biggest issues. And it's really good at combining those resistance genes and stacking them into these resistant biotypes. So that's kind of why the focus was on that. Also, the individuals they were speaking to are kind of from those states that are dealing with water hemp. Pat Tranel is a weed scientist at University of Illinois. And he's doing a lot of this genetic material. I really thought this idea of gene drives was kind of unique. Did anybody look at what gene drives are or want to try to explain that to the group? I'll give you a second. So the gene drives that we talk about, or they talk about in this paper, essentially look to manipulate Mendelian genetics. So based on what we talked about earlier, it's like everything is inherited in a 1-to-1 ratio, and it could be equally distributed. But gene drives essentially bias the inheritance for a specific allele. And so it drives the population artificially quickly to one endpoint on purpose. And in this case, the idea would be to develop this gene drive to get herbicide susceptibility genes back into that crop, or manipulate their seed production genes or something like that, so that it could help to drive that population to become less resistant or less prevalent in the future. But there's a few things limiting the gene drives. I think they listed 5 or 6 things that could potentially be limiting. And those included issues with understanding the target species genes better, how many seeds of the changed individual need to cause the change in biotypes to actually get the population to decrease. What are the implications on other populations? They mentioned the goal of this practice is not to eradicate the species from the face of the planet. It's to get it back under control. And so how is that going to affect the natural biotypes of the species that are seen? What are the regulatory and social concerns? And what are potentially the weeds' ability to adapt to this practice as well? If they can already beat off this herbicide that's going to kill them, how do we know they're not going to get over this relatively quickly as well? So these are all, it was a really neat article because it was kind of like upper pipeline thinking of genetics and manipulating it to improve our management in an agricultural context. Does anybody have any follow-up thoughts before we wrap up that segment of our professional development session? Could you see yourselves using these resources moving forward? A way of integrating these into your classes? Does anybody talk about resistance and frankenweeds in their classes? If you haven't, do you think you might start?
Yeah. I
As a
think
result of this?
it's kind of helpful to talk, when you're talking about, we talk about antibiotic resistance, I think is probably the primary thing that most biology addresses. But I think it's really important to talk about how other things have developed resistance, mosquitoes to different pesticides as well, and then these weeds. So I do think it is a good application for things that are around students. They might see weeds grow. I mean, they might see that more readily than, antibiotic resistance seems to be a little less easy for kids to understand.
Yeah, less visual cues in that
Yeah.
regard, yeah.
Right.
Yeah.
I think one of the examples of gene drives that was recently done was released in mosquitoes.
Yep.
Yeah.
In Florida. And so this is a situation where it is in another species and they're taking it and trying to apply it potentially to weeds as well.
Yeah.
And so I think the mosquito situation will provide a pretty interesting case study to see what ends up happening in that particular scenario. It's not a guarantee it won't cause other problems or other issues in a new system, but it's not the first time this type of thing has been discussed in a natural species population.
Yeah, the social studies aspect is also really interesting to connect with this for sure.
And I know a lot of these resources come from places that aren't necessarily Ohio, but I think that's okay considering most of our participants are not necessarily from Ohio State either. But it is something that we see in the northern states as well. Horseweed is a major one. Horseweed or mare's tail, that's one of the most ubiquitous weeds we have in Ohio, and especially in soybean fields. And most of it is glyphosate resistant. But the main reason why that weed has taken off as far as a resistant population that we see all over the place in Ohio is because soybeans transitioned to no-till production. Horseweed is a very good surface germinator and its seeds are wind dispersed. So it'll produce 100,000 seeds they all have these little wind dispersal mechanisms. They'll be picked up, blown for miles, deposited somewhere else. And if there's no tillage on the ground, they're a surface germinator. And so no-till production soybeans allows them to germinate right on the surface, and then they'll take off. And so that's a case study of Ohio relevant species, not necessarily the pigweeds that they're seeing more down south and out west, but horseweed is one that we have right in our backyards, where a cultural change of agricultural production led to the evolution of this as being a very relevant weed in our production systems.
Yeah, and just another tie-in to the comment about social studies is that if we also consider the module that I presented, open pollinated plants are not as common in agricultural production here across the United States, at least not commercial production across the United States. But because they are less expensive, they are really important for marginal lands, especially in developing countries. So if we look at that cultural connection with the different types of production systems with hybrids versus open pollinated crops, that also has a lot of cultural relevance in that regard.
Yeah, I got my master's degree in weed science, so weeds are kind of, they hold a special place in my heart. Because I like to study them. I also like to make sure they're not a problem, but it's an exciting area of field, and it's one that may not necessarily be well understood, especially by some of our students. Just given because it is a different type of concept, and understanding how that resistance piece ties in, why we should care about it, and what we can do about it moving forward. I think those are really important.
I also think it's a great opportunity for students to understand that we haven't solved all the problems. I always felt like when I taught environmental science that the answer was either quit doing that, totally eliminate whatever it was the problem, or or we tried every solution there was, so there weren't any other options. And I think with this gene drive article particularly, it helps kids to think about, oh, well, there's one way you might be able to do something different. And another one that I was just even thinking about is just the idea that can we make chemistry that now, will address that changed metabolism in those resistant varieties. So there may be other things that, kids will investigate or think about in a different way, because you're saying, hey, this is a problem. We know it's a problem. We've used these management practices. How can we address this problem? And we need new ways of thinking about it. So.
Right. Agriculture as STEM is very much an applied version of a lot of our science concepts. And I think that's the biggest take home. If you take nothing else away from our sessions, it's that agriculture is applying STEM concepts in a very real, practical, and tangible way. Sometimes we think about STEM as being more theory-based and calculation-based and more abstract, but agriculture oftentimes takes a very applied approach, taking those concepts and making a major change or a major difference in people's lives real time. And so it's a really exciting thing for us to be able to bring to you all to share today and have this conversation. And hopefully some of the things we presented today or some modifications of them can make it into your classrooms when you talk about genes or talk about genetic approaches. And again, at the end of each one of these, if we look back at our document there, we can see that they each have kind of learning standards outlined as far as which ones they're addressing from a next generation standards, also from like a state specific version. So for Ohio, for instance, we went through the Ohio standards and try to identify those. So
I think the thing that I keep coming back to, I taught biology for 16 years of the 30 years I taught, and I keep coming back to unfortunately, we don't teach about plants very well, if at all, because it's not addressed in the standards and it's not being tested. So this is another way to get some kids who may be not quite so engaged when you're talking about photosynthesis as a theoretical cellular process, that they might be able to grow plants and see, some of these ways to investigate. And even your chemistry students, starting to think about these chemistry biopathways. I guess the other thing is we're always really teaching students for whatever that comes next. We can't get too mired in the fact that, well, I've got to get through these standards, because that they may or may not care about that. But their next level is where they're, where it's going to be relevant to them. So if you can introduce, hey, there are chemists who are working on these chemistry things, they're geneticists working on these genetic pieces, they're different pathways than I'll be a doctor or I'll be an engineer. So I think it's kind of an interesting way to think about it, and to increase the number of careers we're actually exposing students to. And there's a lot of opportunity in agriculture careers. So right now, the pipeline is only filled by about 60% of the people who come out with an actual ag-related degree. And the other 30 or 40% of the jobs need to be filled by people who may not have an ag background, but are chemists, are biologists, are interested in pathology and things like that. So I do think that there's a real application for our students, the relevance in terms of talking about careers and what are their next steps. And if they like this kind of information, they like doing these kinds of things, and there are really a lot of jobs out there for them. So.
Just for example, here we have our next gen standards that could potentially be addressed by these. May not directly address them, but there are ways to tie them in. Or if you teach these or recognize some of these as standards that you're responsible for, it's a possibility that this could have some fit in what you're doing.